Welcome. I want to introduce you to our interactive web map for visualizing biological data from natural history collections. There are hundreds of natural history collections in the US, and they all store a wealth of specimen data. So for each animal, plant, or other organismal object that these institutions have, there's data on when and where they were collected. Now, these institutions are undergoing a big push for open data to increase accessibility to both researchers and sometimes the general public, and there's also a big push for public outreach, because public awareness often indirectly translates to increased funding for these institutions. There's also an increased emphasis on virtual data exploration that allows researchers to measure traits on biological specimens from afar without having to travel long distances or request loans of biological specimens. Unfortunately, these data are often fairly difficult to access, and they're not that easy to pull large amounts of data. There's also pretty low utility and usability for immediate data exploration or hypothesis generation, and low utility and usability for education. We think that interactive maps may help this. Interactive maps are already widely used to visualize species distribution, visualize movements and migrations, to show how traits are distributed across the landscape. And they're really good for preliminary hypothesis generation without too much big of a time commitment. They're also really engaging and intuitive educational tools. The target users of this type of interactive web map will be researchers in that exploratory phase of data analysis, students in order to give them a larger appreciation and bigger picture of the utility and immense biological diversity that can be explored through natural history specimens, and to citizen scientists to foster an interest in biodiversity and hopefully inspire the next generation of researchers. To ground truth the utility of interactive maps for these type of data, we've taken a subset of data from the natural history collection at the Museum of Southwestern Biology in conjunction with the WIT Lab at the University of New Mexico. We're looking at 4 species that are distributed in the Andes across a wide swath of elevation, and looking at how their blood oxygen transport traits change with elevation. In general, they tend to increase, but these increases may be species specific and may indicate local adaptation. So we're exploring that in an interactive map context.
Thank you, Chauncey. So this is our interactive web application. So here we have our map, which you can zoom in, select each point, which will show you the locality and the elevation. You can sort by the bird age between adult and juvenile. You can also sort between the elevation category of high, mid, and low, and you can do any combination of the 3. Here in this bar chart, you can see the number of specimens in each locality, and they are color-coded by the different elevation categories. And here we have our 4 different types of species from our specimens, and you can filter by not only the elevation locality and the number here, we can also sort by the different species. So here we have our 3 drop downs. Each one contains something different. So our first one shows the hemoglobin levels. So we have box plots and bar charts. These bar charts have a brush on them, so it allows you to select these different concentrations. If we go down here, we have our red blood cell volume. We have a box plot and bar chart with a brush filter. And finally, we have the heart mass, which again has the box plot and a bar chart with a brush filter. And finally, we have our data table here, which shows you the species, sex, age, locality, elevation, the category of elevation, low, high, or mid, and then we have here our hemoglobin. So to compare, we have site 1, site 2. So here we have site 1, which I have selected a low elevation specimen here, and you can see the different heart mass, red blood cell volume, hemoglobin levels. In site 2, we have a high elevation, and you can see these. Currently, this is the only way to compare between 2 sites, but it is an effective way to visualize everything. Lastly, we would like to make some acknowledgements. We would like to acknowledge the Museum of Southwestern Biology at UNM in conjunction with Christopher Witt's lab for our data. We would like to acknowledge those who collected the data within this lab. We would like to thank Dr. Lipping Yang as well as our entire class for our course this semester for their help in creating our web application. We would like to reference the DC box plot example at this GitHub link that we referenced heavily, and we'd also like to acknowledge the untapped example at this GitHub link that we referenced heavily for our web application. Finally, here's our contact information if there are any further questions. Thank you.